Caterpillar-like bacteria crawling in our mouths likely to survive in the oral cavity. Bacteria have evolved to divide along their longitudinal axis without parting from one another. A Silvia Bulgarisi study published in Nature Communications. The division mode of these caterpillar-like bacteria and their evolution from a rod-shaped ancestor. He proposed to establish Neisseria oral bacteria as new model organisms that could help pinpoint new antimicrobial targets. If you are new to our channel, then please subscribe and hit bell icon to get latest scientific information on the go. Although our mouth houses over 700 species of bacteria, and its microbiota is as diverse as that of our gut, not much is known about how oral bacteria grow and divide. The mouth is a tough place to live in for bacteria. The epithelial cells lining the inner surface of the oral cavity are constantly shed, and together with salivary flow, organisms that inhabit this surface will therefore struggle for attachment. It is possible that to better stick to our mouths, bacteria of the family Neisseriaceae have evolved a new way to multiply. Whereas typical rods split transversely and then detach from each other, some commensal Neisseriaceae that live in our mouths, however, attach to the substrate with their tips and divide longitudinally along their long axis. In addition to that, once cell division is completed, they remain attached to one another, forming caterpillar-like filaments. Some cells in the resulting filament also adopt different shapes, possibly to perform specific functions to the benefit of the whole filament. The researchers explain multicellularity makes cooperation between cells possible, for example in the form of division of labor, and may therefore help bacteria to survive nutritional stress. The team of researchers first employed electron microscopy to survey, across the Neisseriaceae family, bacterial cell shapes that include the two standard cell shapes in addition to the caterpillar-like filaments. By comparing their cell shapes and genomes throughout the Neisseriaceae family, the researchers could infer that the multicellular, longitudinally dividing bacteria evolved out of rod-shaped, transversely dividing bacteria. Moreover, they could pinpoint which genes were likely responsible for the unusual multiplication strategy. They then used fluorescence labeling techniques to visualize the progression of cell growth in the multicellular bacteria and finally compared the genetic makeup of these with classic rod-shaped species. Finally, they tried to recreate that evolution by introducing the genetic changes into rod-shaped Neisseriaceae. Although they could not force rod-shaped bacteria to become multicellular, genetic manipulation resulted into longer and thinner cells. We speculate that in the course of evolution, through a reworking of the elongation and division processes, the cell shape changed, perhaps to better thrive in the oral cavity. Apart from helping us to understand how cell shape evolved, multicellular Neisseriaceae may be useful to study how bacteria learn to live attached to the surface of animals, the only place they have been found to occur so far. Half of us is carrying them in our mouths. Expanding the cell biology field to additional morphologies and symbiotic species is also crucial to increase the pool of protein targets, example antibiotic targets, for biopharmaceutical applications. Neisseriaceae can shed light on new, unforeseen protein targets. This information is authentic and purely based on scientific research. SciTips YouTube channel is your daily dose of knowledge, entertainment, and motivation. So don't forget to subscribe and share the video with your friends and family on Facebook and WhatsApp. Thanks for watching.